recording when we talked about rice. Oh, yeah, I've decided I'm not posting the lectures.
The lectures?
Yeah, I'm not posting class.
These?
Yeah.
Why?
So if you want me to pull something back up, I can pull it back up. We can talk about it again at the end, but I'm not posting lectures.
Why record them?
In case somebody's got COVID and they don't email me until tomorrow, then I can send it to them.
Gotcha. But you're not like posting it for everybody.
No.
Okay, fair enough.
Yeah.
Not the worst thing in the world.
Not the worst thing in the world.
Okay. Where is the camera?
I don't know where half our people are.
That's one of them.
About 2 minutes. Not ready yet.
First edition?
Oh, that's what I meant to pull up, and I pulled up the old one. Thank you. Yeah, no, I know. You're like, wait a minute.
Is it that much different?
No, it's really not.
Is it because you think going back and rewatching will be like some color? Like it would be more efficient for me?
Well, I think it's more efficient not to go back and watch them, but to this is what I want. Hold on. Let me pull up the right thing. Then I'm going to talk because I'm not good at doing two things at once. I think you need to practice discussion questions, like where you're integrating the information, not where you're just like, am I memorizing the information? But like, are you assimilating the information and then can generate your own response? Because it's not multiple choice on the exam. It's discussion. Your exams are discussion. So the more you practice discussion, the better you're going to be.
So like the notes.
The notes are good. You should take notes. Absolutely take notes, right? Absolutely look at your notes. Absolutely read the book. But
Notes by itself is not going to suffice.
right. You need to practice doing what you're going to be required to do on the exam. If that makes sense. That's what I want you to spend time doing.
It's the big picture.
Well, right. That's why we have all those discussions. That's why we talk about things. That's why we run this a little bit differently than I've done other classes in the past, right? Okay, so 9 o'clock. Let's see. What's the worst part of planning a party in space? Oh no, I just threw it away. I just gave it away. I did it wrong. What's the worst part of throwing a party in space? You have to plan it. You have to plan it. Oh, that was terrible. Let's see. I have another one. What did the grapes say when they got stepped on? They whined.
Why can't you sleep in space?
I don't know. Why can't you sleep in space?
In space, nobody can dream.
Because it's what?
Because in space, nobody can hear you dream.
Because in space, nobody can hear you dream instead of scream. Yeah, that's good. So don't forget to check in on iClicker if you have not.
Yeah.
Please go ahead and do that. All right. Okay, so this is kind of the end of the end for carbs, right? We're going to talk about carbohydrate metabolism, which technically you already sort of know about, right? What part of carbohydrate metabolism do you already know? Glycolysis, right? So breakdown of glucose. Glucose is a carbohydrate, right? We've talked about formation of glycogen, right? So we've talked about some of it already. So you're going to draw on what you already know, and we're going to add to it. So the big pathway that we're going to talk about in this chapter is the pentose phosphate pathway. So when we talk about the pentose phosphate pathway, we have to kind of change gears, because when we went through glycolysis, what was the goal? What was the name of the game? Make energy, make AGP, make energy carrying molecules, right? Yeah, it took us a while to get there, right? We needed some other pathways to make it happen, but not anymore, not in pentose phosphate pathway. So we call it pentose phosphate pathway, the 3 Ps, right? So in this one, the whole point is not to make energy, right? So what kind of a pathway is glycolysis? Catabolic or anabolic? Catabolic. So pentose phosphate pathway is going to be the opposite. It's going to be anabolic. And so in pentose phosphate pathway, we're going to make two main things. We're going to make NADPH. And the other thing that we want to make is we want to make ribose. We want to make ribose 5-phosphate. Where do you know ribose 5-phosphate from? Precursor for, I feel like we're going to need ribose sugars for something. And RNA, DNA, ATP, NAD+, right? We need it for cofactors and we need it for nucleotides, basically. Cofactors and nucleotides. So we're going to use the ribose 5-phosphate in anabolic pathways where we make things. And then the NADPH is going to be used to basically create a reducing environment so that toxic things can't harm our cells. And we're going to kind of really talk about how that works. But you could say like antioxidants, right? Antioxidants. Helps all of our antioxidants actually work. All right. So the big one's pentose phosphate. Then we'll talk about gluconeogenesis, which will be relatively easy because you know glycolysis. So it'll just be some changes on glycolysis. And then we'll talk about glycogen in a little more detail, but the big one is going to be pentose, right? So there are 3 different names for the pentose phosphate pathway, as if one is not enough, just like the Calvin cycle, right? So pentose refers to what? Number of carbons, and then it's phosphorylated, right? So it is also known as the hexose monophosphate shunt, and we'll find out why that is, or the phosphogluconate pathway, and we're going to find out why it's named 3 different things. So what's the whole point? Well, if you're going to have, let's say you have 1 molecule of glucose 6-phosphate. Where did we get our glucose 6-phosphate? I had a meal, my blood sugar goes up, blood is in the blood, sugar is in the bloodstream, insulin signals the cells to bring in the glucose, but the glucose could slip out. So what does the cell do to it? Phosphorylates it, right? So it puts that negative charge on it. So now it can't just go back and forth dependent upon concentration. It's basically trapped in the cell. So if my ATP levels are low, what happens? I'm starving. I haven't eaten in a long time. I'm going to go through glycolysis, right? That's normal. Okay, but what if I'm not that hungry, right? And what if I need to make things? What if my cells are dividing, my ATP is high? I need to make more DNA. My cells are dividing. So now I need to go in and make the precursors that are required for DNA synthesis or other pathways. So that's where we're going to go into the pentose phosphate pathway. And the point at which the cell decides, am I going glycolysis, am I going pentose phosphate, is that at that glucose 6-phosphate level.
What about whenever we have enough glucose it stores it as glycogen?
I'm going to show you. We're going to talk about each, and then we're going to talk about kind of like all of them together. And okay, these are the different states your body can be in and when. So we will. All right. So you got 1 glucose 6-phosphate molecule. So the first thing that happens is that glucose 6-phosphate molecule is processed, and in that process, we are going to actually reduce 2 NADP plus molecules into 2 NADPH molecules. Now, How is NADP plus different than NAD plus? Structurally, take a guess. What does the P stand for? A phosphate group. So the only difference between NAD plus and NADP plus is that the ribose is phosphorylated. So this has a phosphorylated phosphorylated ribose. It's at C2 if you really want that. But it's the only difference. But it's really important difference. It's a small difference, but it's a really important difference. What if both the anabolic and the catabolic pathways used the same molecule like NAD+? What would you end up having? It would be futile cycling. Nothing would happen, right? So you need different molecules to work in different pathways. So our enzymes are designed to use something that's phosphorylated. How specific are enzymatic pockets? Very, very, very specific. So a phosphate group is a big change. So that allows the enzymes to be specific for the anabolic pathways versus the catabolic pathways. Okay. So we know that we're going to, from one glucose 6-phosphate molecule, we're going to produce 2 NADPHs, excuse me. And we're also going to produce ribose. So our ribose phosphate is going to be that component for our nucleotides, right? DNA, RNA, and it's also going to be our coenzymes. What are some coenzymes? We said ATP, right? What else did we say? Nicotinamide adenine dinucleotide. Do you think that ribose is going to be in that? Sure. Okay, what else? Sure. Very similar structure. Very similar structure. And if NAD plus is on the list, what else should be on the list? NADP plus. And the one that y'all haven't named that's also on the list is acetyl-CoA. So it's really important to keep an idea of where these reactions are happening in the cell. This one's happening in the cytoplasm. So if you look at the overall reaction, right, you're starting with glucose 6-phosphate. And do I show you ending with ribose? No, I do not. That ribose is actually processed into other sugars in yet another carbon shuffle. Carbon shuffle, yeah. Like I know I need that music. I need to be able to just cue that music. I need to have it on my phone or just hit play, right? It just comes up. So what we're going to see is really it's a two-step process. There's an oxidative phase and a non-oxidative phase. And so we'll see it broken down that way, right? But let's start with our oxidative phase, right? Our oxidative phase only has a couple of reactions. The non-oxidative phase is where you're going to do a ton of carbon shuffling, a ton. So not that you have to memorize every single step in the non-oxidative. The steps in oxidative are really important. The non-oxidative, we're just going to kind of talk about generally. And you have to kind of generally know what enzymes are required. So if you look at the pentose phosphate pathway, right, we know that we need NADPH kind of generally in our cells, but depending on what cell type you are, you need a different amount of NADPH. So things like the red blood cells need a really high level of NADPH, right? Because they're carrying oxygen. You can make superoxides. You can make all kinds of crazy radicals and things like that, that you need a high level of NADPH in order to reduce those crazy molecules, right? You also need to know how much DNA are you trying to make? How much RNA are you trying to make? How active is that cell? that is going to be determined by your nucleotide pools. If your nucleotide pools get low, then you're going to start shunting glucose away from glycolysis, and you're going to start using it in pentose phosphate, right? The other thing is if ATP levels are low, right? If ATP is really low, we're going to turn off pentose phosphate and we're going to get shunted over to glycolysis, right? So you got to kind of think about them. And we're going to look at each of the 3 metabolic states in a pathway, which I think is kind of easier to look at in just a second. So look at your structure, right? We look over here. So this is nicotinamide adenine dinucleotide phosphate. So here's our ribose sugar. And if you look at C2, we're phosphorylated. So you know that this must be NADP, right? NADP plus. So what ends up happening is that this can act as an electron carrier, right? So it can pick up those electrons, and we are reducing the molecule to NADPH, right? So what parts of this molecule are really important? It's just kind of important. You don't have to be able to draw it, but you have to know what are the individual components, right? You need riboses inside of it. And that's really important because you need to know when the pentose phosphate pathway is going to be activated and when it's not. So if you know what the components are of the different requirements of the cell, you can know when this pathway is going to be activated or not, right? So if we're going to make ribose sugars in this pathway, if we need NADPH, we're going to have to activate the pentose phosphate pathway, right? Because not only do we need the NADPH, we also need NADP plus, right? With me so far? Yeah. Okay. So here's our 3 different states. This is kind of what we talked about in general, right? If you look over here, here's glucose. So look kind of towards the bottom. What happens, the easiest one, the absolute easiest one is here. What happens if ATP is low? This is our first energy. It's not really our first. Which one was it? It was the 3rd one on the previous slide. What happens if ATP is low? It goes through glycolysis. So all of this is going to be shut off. This whole big old crazy pathway is going to be shut off, and you're going to promote glycolysis. So if ATP is low, we shut down the pentose phosphate pathway and we upregulate glycolysis. All right. Okay. Now, what's the next one? Let's say nucleotide levels are low. Right? Our cells growing and growing and growing, and we're running out of the nucleotides that are required to create new DNA. So what do you need in order to make nucleotides? What kind of sugar? Ribose. You need ribose. So most of the ribose 5-phosphate is going to be used to make nucleotides. So we know part of the pentose phosphate pathway, we have this oxidative phase, then we have the non-oxidative phase, that we're going to go in the oxidative phase from glucose 6-phosphate, and we're going to make ribulose. The difference between ribulose and ribose is pretty easy. It's just an isomerase that's required to convert it. So we don't have to do a whole lot of carbon shuffling if we want to make nucleotides. All we do is go through the oxidative phase, get our ribulose, and then go through an isomerase and make our ribose, right? Okay, now our third really was our first. What if we have low levels of NADPH? We're going through an oxidative stress in the cell, right? And so all of our NADPH is being depleted. We want to upregulate pentose phosphate. So we're going to upregulate pentose phosphate pathway. Do we really care about the non-oxidative part of that? No, we really don't. Here what's really important is the oxidative phase. So if you look at this, I could pull glucose that was destined for glycolysis, absolutely. But what if I still need to make ATP? Is there another way to get glucose? You could break down glycogen. You could break down, what else? Well, you could break down fats, right? There are different sources of glucose within the cell. So you gotta just kind of keep that in the back of your mind, that not just glucose that's in the bloodstream can be used in this pathway, right? You can get it from other sources. So when you look at the pathway, right, what I want you to see is in the oxidative phase, we have to start with glucose. And we're going to get ribulose, and we can go in and make that ribulose basically into whatever sugars we really want. But in order to make this happen, you actually, right, are going to start with ribulose, but then you can go through all kinds of carbon shuffles, and you're going to end up with these two different sugar molecules, glyceraldehyde 3-phosphate and glucose 6-phosphate. Now, what we can do with these two is we can actually use an isomerase and turn it back into what? Glucose. So we can use these sugars that are made in the pentose phosphate pathway to remake our starting material. We're not breaking down the sugar completely. That's not the point of the pentose phosphate pathway. So this can't happen in something like glycolysis, right? It does happen a little bit in citrate, right? Because our product becomes our substrate for the first reaction. So it's kind of like that. Where here we have some end products that can then be used to regenerate our starting material. So if all you need is the oxidative phase, you're not replicating your DNA a ton. You just really need the antioxidant capabilities of the pentose phosphate pathway. You can use the sugars that are produced in the end to regenerate the beginning. Yeah.
Sorting glycolysis, the thing that drives that is the ATP, but in this, it's just that redox reaction.
Well, what drives this is going to be the ratio of NADH and NADPH and NADP plus. So just like our ratios of ATP and ADP, right? So you do have a little bit of energy charge dependency, but a lot of this is really driven by ratio of NADP plus and NADPH. Okay, so let's start at the beginning. Let's start at the oxidative phase. So our oxidative phase is only 3 reactions. So we start with glucose 6-phosphate. And what we're going to do is we're going to go through an irreversible reaction in the very beginning. How do I know that this is the very first one right here is going to be irreversible? It has a very negative delta G, right? So we're giving off lots of free energy. So this is irreversible. Irreversible, right? We're committed. The other reason that it is irreversible is this product here, 6-phosphoglucanolactonone. This has no other metabolic fate in the cell. No other metabolic fate. What did we produce in this reaction other than this sugar? What else did we produce that's really important? Here's our first reaction where we're making one of those NADPH molecules. So we still have a closed chain, right? We still have a cyclic sugar. So our second reaction is going to cleave open that ring structure, and we're going to get 6-phosphoglucanate. We don't make an NADPH from this one, but we do need that sugar linear in order to do all the other reactions. So this first one, glucose 6-phosphate dehydrogenase, I want to tell you, glucose 6-phosphate dehydrogenase. You're going to see this G6PD. People abbreviate this enzyme. Remember, like PFK1, right? You're going to see this abbreviation over and over and over and over again. So you need to recognize that.
Abbreviations for the substrate, for the product?
What about what?
What about the abbreviations for the product? So like for 6-phosphoglucose,
Lactinone.
they just call it.
Yeah. People don't talk about this substrate so much. This one's, so it's not, but this enzyme is super important, right? This is your regulatory point in the pathway. So people talk about the enzyme way more than they talk about the product. Just one of those things. All right. So now we've got a linear product, right? Because we've gone through lactinase. So we're just going to cleave our ring. Now we're going to go through another reaction. What are the two things that happen in this reaction to our 6-phosphogluconate? It has to lose a carbon, so it's a D. It's a decarboxylase reaction, right? Because here we had our sugar. But they're both also, if you look, we have a dehydrogenase and a dehydrogenase. Both of the dehydrogenases produce NADPH. So this is what kind of a reaction? Well, you're oxidizing is what I'm just looking for. OXID. Because you have a dehydrogenase that's making NADPH. Okay, so now we've made this ribulose 5-phosphate. Now we can go and use this sugar to do a couple of different things. But it's really important to keep track that you've lost a carbon in here. So we started with how many carbons? We ended with how many carbons? 5. Okay, so now we're going to do the carbon shuffle. Yeah, so when we talk about why pentose phosphate pathway is so important, right? We said you're going to either use it for biosynthesis or you're going to use it to reduce dangerous things inside of the cell. So do you know anybody who can't eat fava beans? Always makes me think of what's the movie Silence of the Lambs, fava beans, right? Yes, no, nobody's. So if you have a mutation in the G6PD, what's going to happen is your body is going to be unable to handle situations where you have highly reactive species. So for example, malaria, right? Malaria is an organism that will infect red blood cells, right? Now, do you remember when we did our things about anemia, right? And if you had sickle cell anemia, you were resistant. Do you remember this? So this is a similar sort of deal where we have a mutation where it's bad. It's actually bad. So if you have people who have a G6PD mutation, they're usually symptom-free until something stresses them. Then they have that stressor and they can't handle it, and the red blood cells burst because they don't have enough NADPH in order to create a reducing environment, and it's very, very dangerous. The same thing happens when people who have this G6PD mutation, they live their lives normally, they eat fava beans, and fava beans produce something that is highly reactive, and if you don't have a reducing agent around to reduce it, it damages your red blood cells. And so this was how they found this mutation. It was people who were like perfectly normal, and fava beans are really big in Mediterranean diets, right? So people would come into the hospital after eating fava beans and having these huge reactions. And so they were able to take the DNA and figure out where the mutation was, and that this is the problem, right? If you don't have glucose 6-phosphate, are you doing the oxidative phase of the pentose phosphate pathway? No. So what happens to NADPH levels? So why is it so bad to have low levels of NADPH? Well, what does NADPH actually do? NADPH will regenerate glutathione, reduce glutathione. So you have two forms of glutathione, the oxidized form and the reduced form. So in the oxidized form, you have these two sulfur groups that are coming off of what amino acid? Cysteine. Cysteine. So in the presence of NADPH, we can reduce oxidized glutathione to the reduced version using glutathione reductase. Glutathione reductase requires NADPH. If you don't have NADPH, glutathione reductase can't work. Therefore, you don't make any reduced glutathione. If glutathione is reduced, right, now these sulfhydryls can actually pick up electrons. That's the great thing about them. So watch what happens. Okay. Right, so this is what we said. We have glucose 6-phosphate. We're going to go through pentose phosphate pathway, right, via G6PD. We're going to create our products, and that pathway is going to keep going, right? So you could just do this, right? This is the rest of the pentose phosphate pathway. But the important part is that we make NADPH. The NADPH is used to convert our oxidized glutathione into reduced glutathione. That reduced glutathione can take things like hydrogen peroxide and convert it into water. Is hydrogen peroxide dangerous? Yeah, absolutely, right? We put that on our open wounds in order to kill everything that's in there, and you see it bubbling up, and yes, it kills things, right? It's very, very bad. So you can have things like that inside of the cell because you have extra electrons from the Q cycle, right? That get picked up by your oxygen molecules that are produced in that are not produced, that are around when you're doing cellular respiration. And in the presence of hydrogen ions and that extra electron, you're going to start making some hydrogen peroxide. So you have to have a way to detoxify the hydrogen peroxide that's sitting around on the inside of your cell. So that's what glutathione peroxidase does. If you have a G6PD mutation. Yeah. One of the,
We used to take away that this semester.
yeah, it was really cool. Yeah, so one of the case studies we did in the previous semester was about the G6PD mutation, and it was really cool. It was really cool. But you know, you can only do so many every semester, and everything's really cool, right? So, all right. So now we've done our oxidative phase, right? We have produced what sugar? Ribulose 5-phosphate. So here, these are all ribulose 5-phosphates. So how many carbons in here? We have 5 carbons in each ribulose 5-phosphate molecule. Now we're going to go through and we're going to use an epimerase. What does an epimerase do? Not necessarily not the anomeric carbon. It switches the hydrogen and the hydroxyl on any carbon that is chiral, right? So when you're drawing all these structures, right? If you have a perfect T, a lot of the times they do it like this, right? Here's your carbon that's going to turn into your anomeric carbon, right? And then they do them like this, right? You've seen that? So anywhere where you have a perfect point, you have a chiral carbon. What's a chiral carbon? 4 different groups attached to the side chains, right? So they can't just flip-flop on their own. They can't just rotate and switch the positions of those four. You would actually have to make and break bonds, right? So that's what epimerases do. So if you have something, for example, on ribose, if you look right here, right? What you had was an OH on one side and an H here. And when you go through the epimerase, it's the opposite. Do you see that in the second molecule in the xylulose? Yeah, so that's what the epimers are going to do. So are you going to change the number of carbons? No, you're not. Now, so the types of enzymes in our carbon shuffle, we're going to have epimerases. Now, we actually have to then change the number of carbons. So if you look from your xylulose, and we're going to go to two different things. We're going to go to our glyceraldehyde 3-phosphate. When we do that, we're going to go through a trans-ketolase. So we're going to make something that's 3 and something that, look at your top product, how many carbons do we have? And the septo, hepto, 7. We have 7. So 7 and 3, right? So what have we actually done? We've actually transferred carbon units. So that's a trans. I'm going to show you. Ketolase. Okay. So we have trans ketolases. Now what's going to happen again is go to erythrose and go to fructose. We change numbers of carbons again, right? So now we have to have an enzyme that's going to convert. We go from, how many's here? 7. Now we have a 4 and a 6. Okay, so that's going to be our trans aldolase. LDOLAS. And then we can convert some of our products here, the fructose 6-phosphate, into glyceraldehyde and xylulose. But what we really want, right? What are the ones that we're super interested in? We want the 2-glyceraldehyde. We want the fructose 6-phosphate and the fructose 6-phosphate. Those are our really important ones, right? Okay, so those are the ones that we can use to do what? What can we do with these now? Well, what if, so let's just say we're under a really stressed situation, right? We've eaten fava beans and we don't have a mutation. What do we want to make more of? NADPH. So what are we going to do? We're going to take these sugars and regenerate what? Glucose 6-phosphate, so that we can go through the oxidative phase of pentose phosphate pathway again. All right. Okay. So if we look at the difference of our two major enzymes, our trans-ketolases and our trans-aldolases, right? So if you look at a trans-ketolase, how many carbons are you actually moving? Right? If you start with ribose, ribose has how many carbons? 5 carbons, right? Xylulose is 5 carbons, right? We're going to turn that into a glyceraldehyde, which is 3 and a 7. So what's going to happen? We're going to take 2 from the xylulose and plunk it on top, right? So a trans-ketolase is going to transfer 2 carbons, or a 2-carbon unit, however you want to think about that. So we had an aldehyde in ribose. I can speak today. You had a aldehyde, right? What's an aldehyde? Y'all remember aldehydes and ketones? Yeah. So this is how I remember aldehydes and ketones. Aldehydes are all alone. Ketones, you need 2 things like a lock and a key. So if you look at ribose, if you look on your carbonyl carbon, right? You have 1 R group and a hydrogen. So it's all alone, so it's an aldehyde. But then once we go through this transketolase reaction, now I have, this is an R group. Whoa. Why am I circling that guy? This is an R group. This is an R group. So that's a ketone. Yeah, lock and key, me too. Okay, so then look at your transaldolase. How many carbons are you moving? You're going to move 3 carbons. Yeah. So that's the difference between transketolase and a transaldolase. It's just the number of carbons that get moved. That's it. So that's how we start our carbon shuffle. Okay. So this carbon shuffle reminds you of what other reaction we briefly talked about. Calvin cycles. Calvin cycle in photosynthesis, right? Okay. So now what's going to happen? So if you look at what you're starting with, right? You're going to start with our glucose 6-phosphate, right? So what's going to happen in oxidative is that we're going to make our ribulose 5-phosphate. So if you look down here at the bottom at B, once I'm finished with oxidative phase, I'm going to end up with ribulose 5-phosphate. And if I go through the non-oxidative phases, I can end up back at what I started with the glucose 6-phosphate, right? So it does replenish it, but does it replenish it equally? Like if I made 6 of my ribulose 5-phosphate molecules in oxidative phase, am I going to make 6 glucose 6-phosphates? I can't, right? Why? I don't have enough carbons, right? I don't have enough carbons. Ribulose 5-phosphate is a 5-carbon molecule. If I start out with 6 5-carbon molecules, I can only end up with 5 6-carbon molecules. Yeah. So you have to kind of think about, why are we shuffling all these carbons? We're shuffling all these carbons so that we can get the exact same starting material. But you can't pull carbons out of thin air, right? We're not plants. We can't pull carbon and integrate it into substances, right? It would be nice. It would be nice, right? Yeah. Okay. So in the last pathway, we had what 2 components did we circle that we had? Fructose 6-phosphate and G3P, glyceraldehyde 3-phosphate. So if we pick up with those, with our fructose 6-phosphate and our glyceraldehyde 3-phosphate, you're going to go through either phosphoglucoisomerase to make glucose 6-phosphate, or if you're the glyceraldehyde 3-phosphate, you'll go through an aldolase. Then you go through fructose bisphosphatase 1. So fructose bisphosphatase 1. What does that kind of remind you of? Like it should, something in glycolysis, right? What in glycolysis? So think about when you want glycolysis to happen and when you don't want glycolysis to happen, right? If we're going through pentose phosphate pathway, we've shunted things away from glycolysis. So this fructose bisphosphatase 1, we're converting fructose 1,6-bisphosphate to fructose 6-phosphate. Is that a common reaction that occurs in glycolysis? It does. It's an inhibitor of the glycolytic enzymes, right? So what we actually want is we want this reaction to occur in pentose phosphate, right? So we have to have a different enzyme so that we can go in the opposite direction of glycolysis. right? So what you'll see is that the things that inhibit like the glycolytic enzyme are going to be promoters for this enzyme. This enzyme is also in gluconeogenesis. So you'll see that they're oppositely regulated.
So you're saying Pentose wants to
No, the product.
Oh,
Product. So you might want to go back and like refresh yourself on regulation of glycolysis, because that's going to be really important for gluconeogenesis too. That's not going to be a bad thing to go and look up. So, right, if we're talking about the big pathways in general, we've kind of already said this, but it's nice to kind of put it in one big picture, right? If you have lots of NADPH, do you want to go through pentose phosphate pathway? No, so you're going to basically shut down the pentose phosphate pathway, not completely, but you're going to inhibit it mostly. Remember we said things aren't really on completely and off completely. You're going to promote going through glycolysis, and you're going to promote making ATP, right? So if you have high levels of NADPH, NADPH is actually going to bind to your G6PD. What is G6PD? What does that do? That's our very first enzyme in the pentose phosphate pathway, right? It's our very first enzyme. That's the one that's going to start producing NADPH. So it's actually going to compete with the NADP plus for binding, right? In order to make glucose 6-phosphate dehydrogenase work, we need NADP plus. The NADP plus has to bind to the glucose 6-phosphate dehydrogenase. So what's going to happen here is you've got lots of NADPH that's going to outcompete the NADP plus and inhibit glucose 6-phosphate dehydrogenase. Okay, so then our other condition over here, we've got lots of NADP plus. Do we really want to go through glycolysis? No, that's saying that we really need our reduction potential of our cell to go up. So we're going to inhibit glycolysis. We're going to go through pentose phosphate pathway. Once our NADPH gets high enough, then we're going to come back and inhibit the pentose phosphate pathway again. Right? It's all that feedback between the two. It's going to be super, super important. So generally, what would we call this shift in the cell? Are you creating things, or are you destroying things? You are breaking them down, so we call that, this is catabolic, and this one is anabolic. Okay. All right. So, gluconeogenesis. Before we talk about gluconeogenesis, what I want y'all to do is to take out a sheet of paper or whatever, your iPad, I don't care. You're not turning it in, just like start a fresh page somewhere. Do you remember glycolysis? Try, without looking, to write out glycolysis, then talk to each other, see if you can fill in some more gaps, and any gaps that you have, go to the book. So right now, try to write glycolysis. Whatever you know, put it down. Anything, starting material, ending material, anything. We said the first step, right? So you start out with something in your bloodstream and then you got to trap it in the cell. Wait, say it again. Just write what you know. It's okay. Just write what you know. You're gonna be able to look it up in just a minute. Once you get stuck, turn to somebody else. Say what you got. If you're both stuck, go look it up. But I bet you, as you get rolling and you start talking, I bet you're gonna remember more than you think. If you need to go look it up, you can go look it up. But come and talk about it. And see if you can remember the big enzymes in glycolysis. Because those are going to be really important. So as you're drawing this pathway, can it go in reverse? How are you drawing your arrows? Are you drawing them all as down arrows?
Some of them are reversible, some of them are reversible.
Some of them are reversible, some of them aren't. See if you can Draw your arrows so that you know which ones are reversible and which ones are irreversible. Because that's a key thing. Right, remember how we did that big thing about is photosynthesis the opposite of cellular respiration? And what we came up was not really, no,
Not really.
not really. So that's what we're gonna come up with gluconeogenesis and glycolysis. I'll give you a sneak peek. They're not the opposites. They're not the perfect opposites. How many reactions did you get? 13.
There are 10 in all.
Oh, there are 10 in all. That makes so much more sense.
Where did the other 3 come from?
It's new.
What does it mean?
Getting close, y'all? And how many ATP are you going to make? Only 2. Only 2.
That's outside.
That is outside of the mitochondria.
Most of those states. Of course, there are differences between cells.
Like 2 more minutes, you think? Yeah. All right, so y'all ready? Just about close. All right, so when you look at your glucose, or you go look at your glycolysis pathway, right? Do you end up with a sugar at the end? You have a sugar at the end. No, right? Okay. So gluconeogenesis is the reverse, right? We're going to start with a non-carbohydrate component and turn it into glucose. So this is the opposite. Do we have the same number of reactions in glycolysis as in gluconeogenesis? How many did you get? 10. How many in gluconeogenesis? 11. Oh, okay. So one of the steps in gluconeogenesis is going to take 2, where it took only 1 in glycolysis. So gluconeogenesis is only when your dietary sources of glucose are low, except you have an organ that is very picky about what it eats. The brain. The brain wants glucose and glucose only. So a lot of times what happens is to feed the brain, the liver and the kidneys will use gluconeogenesis in order to make the glucose that the brain wants, right? The brain's pretty picky, or brain. So it occurs mostly in the cytoplasm, except that you have one enzyme that occurs that is active in the mitochondria, and that's pyruvate carboxylase. So remember, it is the opposite of glycolysis in that gluconeogenesis is anabolic, whereas glycolysis is catabolic. Are we going to get an opposite amount of energy from glycolysis and gluconeogenesis? Y'all just told me that you get 2 molecules of ATP from glycolysis. Are we going to spend only 2 molecules of ATP in gluconeogenesis? No, how many are we going to spend? Be careful. Really, it's 6. Really, it's 6 because it requires a GTP, but a GTP is basically an ATP equivalent, right? So we're going to start with pyruvate. In order to run gluconeogenesis, what else do we need? NADH. NADH. We're going to use NADH. We're going to make in the end glucose, NAD plus, and then our byproducts of ATP hydrolysis, right? Okay, so before I look at this, so the glycolytic enzymes, which ones were the really important ones? Which ones were the irreversible, really big negative delta G ones? Start at the beginning. Hexakinase. PFK, this one's PF. PF. K1 and pyruvate kinase. Okay, so these are our big 3 negative delta Gs. So when you look at the gluconeogenic pathway, you have shared reactions with what? We call these shared reactions. What are they shared with? Glycolysis, because they have a delta G value that's really close to what? 0. So everything that you learned previously is going to work to your benefit because you already know most of the reactions that happen. But these that have very high negative delta G values in this last column, can we just run them backwards? No. So what we need to have are other enzymes that are going to make those reactions happen in the reverse, but make them happen in the reverse with high negative delta G values, right? So we have either shared reactions, or we call these bypass reactions. So we need a bypass reaction for pyruvate kinase, for PFK1, and for hexakinase. Okay. So where do we get our extra enzymatic reaction from? If you look at hexakinase, you have A kinase is something that phosphorylates. What's something that dephosphorylates? A phosphatase. So kinase and a phosphatase are two opposites, right? One phosphorylates, one dephosphorylates. So you have a glucose 6 phosphatase that does the opposite of a hexakinase, right? So instead of adding a phosphate, you are removing a phosphate. Okay, phosphofructokinase 1, we now have fructose 1,6 bisphosphatase. That's another phosphatase. Okay, now that's 1 for 1. What happens up here? Where we had one enzyme, pyruvate kinase, now we have two, a pyruvate carboxylase and then a phosphoenol pyruvate carboxy kinase. So now that we kind of understand, okay, what were my glycolytic enzymes? Now, how is the pathway different in gluconeogenesis? Now let's kind of go back and look at what happens. Y'all good on that? So was it good to learn glycolysis before? Okay. So your body needs glucose, right? Your cells need energy. And this is true in animal cells. This is true in plant cells, right? So plant cells have to be able to do gluconeogenesis as well. So when we look at this, there are different places where you can get the precursors to make glucose, right? So if you are a plant, what are you going to do? You're going to use CO2 and you're going to fix the CO2. So this is for plants only. Plants. And you're going to convert it into sugars. You're going to start with glyceraldehyde 3-phosphate, then you're going to go up fructose 1,6 bisphosphate to fructose 6-phosphate to glucose 6-phosphate to glucose. Because once you're dephosphorylated and you're in simple glucose, this is going to be exported out of the cell. Exported out, right? Which is great, which you want. Except for in muscles. So I'm going to tell you in muscles, this reaction doesn't happen. So in muscles, you don't remove last phosphate. You want to keep it trapped. Your muscles know they're going to need the energy, so they're going to keep it trapped. Now the liver is different. So this pathway is really for the liver. This pathway is really for the liver. A lot of times that's what we do the comparison between muscles and liver, muscles and liver. Okay. So we're in a situation where we're in an animal cell. We don't have blood glucose. Our blood glucose has dropped, right? What are we going to do? Preferentially. Let's say we have oxygen around. What are we going to do? I still want to go through what? Okay, so I have low blood glucose, but I'm a typical normal, not starvation mode, right? But I just don't have glucose hanging out in my bloodstream. My brain is hungry. What am I going to do to feed my brain? I could break down glycogen. I could absolutely break down glycogen. Where else can I get glucose from? We're gonna assume that our glycogen storage is depleted. We're gonna go to fatty acids. So we go from fatty acids, we get glycerol, right? And then we can go back up, we can make glucose and export it out. Okay, now I'm stuck in a cave. I haven't seen the light of day in 2 months. Do I have any fat stores left? No, I'm out. I am out. What am I going to do? I'm going to break down muscles. So do you see here where we say nutrient limitation? only under starvation conditions are you actually going to break down your own muscle tissue to generate amino acids to enter your gluconeogenic cycle. Now, what if I need the glucose, but I don't have oxygen around? I'm not necessarily in starvation conditions, but I don't have oxygen. So like my muscles are working out, right? In an anaerobic way, like weightlifting, not, you know, running, but like weightlifting. Then I'm going to go through anaerobic respiration. I'll make lactate, and lactate can go in and be used to create glucose, right? So that's why when you see like sprinters and things like that, when they run, or really, really long distance runners, do they stop immediately when they're done with their race? No. Why do they keep walking, keep moving, keep going? Because they've piled up lactate inside of their muscles. And so if they keep moving, what they're actually going to do is process that lactate and get rid of it, right? But if they stop automatically, then you'll end up with muscle cramps because of all this lactate, all that lactate buildup. That's why they keep walking. Don't just stop, keep walking. Yeah, absolutely, absolutely. Okay, so let's look at gluconeogenesis. Gosh, that's small. So if you're looking at gluconeogenesis, right, you got to start at the bottom. What are our 4 big enzymes, right? In glycolysis, we had 3. In gluconeogenesis, we have 4. So in the very beginning, do you remember that we had 2 enzymes that took the place of pyruvate kinase, right? So our pyruvate carboxylase and our phosphoenol pyruvate carboxykinase. These are going to take the place, right? We're converting here pyruvate first to oxaloacetate. Then we are going to convert oxaloacetate into phosphoenol pyruvate. This is going to take an input of quite a bit of energy. What are you investing in this phase? 1 ATP and 1 GTP for an equivalent of 2 ATP, right? So that's a big investment right there. Right? Now, the reactions going up, do you see how they're double-sided? Can you start adding to the diagram that you just made of glycolysis to make a pathway for gluconeogenesis? So, yeah, so what I do suggest you do is take your own hand-drawn glycolysis and add in a different color, in a different whatever, your gluconeogenesis, right? Where is it different? Because if you just try to memorize gluconeogenesis, it doesn't quite make sense. But if you compare it to glycolysis, it's going to make perfect sense. So do you see how these are all one-way arrows? Pyruvate kinase is a one-way arrow. What happens when we get to the shared reactions? They're all what? Double arrows. They're all double arrows, double arrows, double arrows. Oh. Phosphofructokinase 1, right, is an irreversible reaction, so we can't just go backwards. So instead, we're going to use a different enzyme, fructose 1,6-bisphosphatase, right? So this one, now what are we doing? Here, we went from fructose 6-phosphate, we phosphorylated it and got fructose 1,6-bisphosphate. So what are we doing when we go in reverse? We're removing a phosphate group, so we are dephosphorylating. So now we have fructose 6-phosphate, but we don't want fructose 6-phosphate, we want glucose. So we convert that, but that's a reversible reaction, so that's okay. But now we have glucose 6-phosphate. If you're in the muscle, you're done. If you're in the liver, we need to remove another phosphate group. So instead of having hexokinase phosphorylated to keep it in, now we have glucose 6-phosphatase that's removing the phosphate group.
But these are the bypass reactions.
These,
11, 9, 2, and 3.
yep, these are all bypass reactions. Yeah.
The enzymes all same names?
Yeah.
Like the byproduct.
The shared reactions, you mean? Yes.
They were named, really from glycolysis.
Well, yeah, glycolysis was biochemically, historically figured out first. So that's where the name came from. Yeah. Yes, absolutely. It's a good thing.
The phosphorylated kinase instead of transferring the inorganic phosphate to the substrate, it would transfer it to the base. Like in the opposite.
It'll transfer it to something else. It won't make ATP because you're not generating ATP from this. This is requiring energy, right? So you're not making it, but you are transferring it off, right? So, but you need those inorganic phosphates because that's what you use as substrates when you go through electron transport and ATP synthase, right? So you make them in the cytosol, they got to be transported in, right? You're starting to see all the connections between our previous pathways. Yeah. You're not making ATP. Yeah, be careful about that. Where just because you used an ATP to phosphorylate something, when you go through gluconeogenesis and you do the reverse, you are not making ATP. Right?
So the point is to get the product for ATP.
The point is to make glucose, and usually like in the liver, it's exported out. So if we're in the liver, right? We're in the liver. We've depleted glycogen, but our brain is hungry. Glycogen depleted, but brain hungry. Right? So we can't just break down glycogen and give the brain the glycogen stores. So what do? We go through gluconeogenesis and we create glucose that gets transported into the blood and goes to the brain. Yeah. Yeah.
So in the body, all of the processes are going on like gluconeogenesis and breaking down glycogen.
They can all happen theoretically simultaneously. They typically do not. Typically, one thing's up, another thing's down, right? Typically, you have seesaw relationships between a lot of these pathways. So they're technically not all going at the same time.
But those three are kinds.
Yes.
So let's
Yes.
say that
Let's say that pentose phosphate pathway is like
Pentose phosphate pathway is what?
I'm don't worry about me. I'll ask you later.
Okay, write it down. As you're thinking of it, write it down.
Well, some of them I'm trying to. I know what I need. I'm struggling with words.
Okay. So if we look a little bit closer at some of our really important gluconeogenic enzymes, right? Look at the very beginning. We're going to start at pyruvate. Pyruvate has how many carbons? 3 carbons. So we're going to have to build our carbon molecules, right? So our very first reaction these are right bypass reactions, right? We're actually going to need to invest in ATP. So we're going to use energy in order to add a carbon group. Where does our carbon group come from? Where does HCO3 minus? What's it? Bicarbonate. So in the presence of biotin and ATP, pyruvate carboxylase can take that carbon containing group and put it onto pyruvate. Now, got to think about regulation as we're going through all of these pathways. If you have a low energy charge of the cell, are you going to bother going through gluconeogenesis from pyruvate? So come back, right? So if you have a low energy charge of the cell, are you going to take pyruvate and be able to go up and make gluconeogenesis. No, you're not. So having a lot of ADP around is going to be an inhibitor of this reaction, right? Now, think about acetyl-CoA. If you have a lot of acetyl-CoA, you can what? You're what? You're not making ATP?
No, the creation of these, it's no makes it.
So you can use acetyl-CoA in order to make ATP when we're talking about glycolysis and then citrate, right? You're going to use up acetyl-CoA. So if you have a lot of acetyl-CoA, do you want to go through glycolysis anymore? No, but now you can go through gluconeogenesis. So it's a positive regulator. Okay, so now we have our 4 carbon containing molecule here. We're going to invest another little bit of energy. And what we're going to do is we're going to lose one of those carbons, but we're going to phosphorylate our 3 carbon group, right? And in the process, we lose that carbon as carbon dioxide. So sometimes you're going to invest carbons only to lose them a second later. But you need this molecule so that you can use all of those enzymes that were in glycolysis as reversible reactions. Because if you don't have the right substrate, you can't use those enzymes, right? So we have to create the substrate that can be used in the reversible reactions of glycolysis. So we've already invested basically 2 ATP equivalents in the very first step. It's really 2 steps, but sometimes we call it 1 step because in glycolysis it's 1 step, right? Okay, are we time? Yeah, okay. We'll pick up on biotin next time. But go through and study your glycolysis and look and see if you can add gluconeogenesis to your glycolysis, right? So you can really understand what it's bypassing, where it's bypassing, what's happening. What's that?
Update the model.
Right, update your glycolytic model so that you can include gluconeogenesis.
Oh, no, I know.
Make sure you've checked in on iClicker.
Tuesday next week or Thursday?
Tuesday, I believe Tuesday.
Because like I said.
No, Thursday.
Thursday?
Thursday.
All right, cool. Because like I was asking, it said like
No, Thursday.
Thursday.
Thursday. That gives me more time. That's good. Did you hear that? It was Tuesday. We were talking about Tuesday, but it's Thursday. It's Thursday. Yeah. Yeah. You want to see if you check in?
I was gonna say, I don't even know how to check it.
So you need to download
I got the
the,
iClicker thing, but it only pops up my old organic.
so you need to add a class. You don't have to pay for it because we're not doing like clicker questions.
Of course. So I'm just guessing biochem too.
Yep. And you should show up here as present.
One of the days I came and I didn't know how to use it.
Hunter, yeah. I know you were here one other day.
Okay.
Yeah. Okay, so yeah, you're on the list. You're good.
Awesome, thank you.
Yeah. Oh, it's still on Zoom.